Okay, we are moving forward to 8.5, which is the process of translation. It's the final process in converting a mRNA message that was made in transcription into a polypeptide or a protein. So, if you recall, in the process of transcription, the mRNA picked up the message from the nucleus and took it out into the cytoplasm for the final process of um, this whole DNA to a protein, which is going to be translation. What translation is going to do, it's going to convert that message into proteins. And there are specific codes that are going to bring or code for an amino acid. So a codon, this is important to write in your notes. A codon is a sequence of 3 nucleotides. You can see in the picture given. Here's your segment of mRNA. 3 nucleotides that will code for a specific amino acid. When you put many amino acids together, you get a polypeptide or a protein that is going to go and do something within your body. So a codon is found on the mRNA strand, and it's a sequence of 3 nucleotides. When we are matching a codon to an amino acid, we can determine what that amino acid is going to be by looking at those 3 letters. This table is going to help you determine what that amino acid is. So if you have, in the example given, find the first base, which is going to be C in the left column. So you're going to go on the very left side of this table, find C. Step 2 is to find the second base, A, which is in the top row. So if you go from 1, which is C, over to A, which is the top, which is the second base, and then you're going to find the third base, U, in the right column. So you go left column, top, right column. So if we go C, A, U, we can see that CAU codes for histidine, abbreviated as HIS. That specific amino acid will build with other amino acids to form a protein. We'll do a little bit of practice with this, determining and finding specific amino acids. But basically, what you're going to have to do is take this table and you're going to have to determine what amino acid we are asking for if we were to give you a codon. Remember, the codon is found on your mRNA strand. A change in the order in which codons are read is going to change the resulting protein. You can see in the picture, the reading frame 1, CGA codes for the amino acid arginine. And if you were to move that reading frame in reading frame 2, you would get ASP, which is aspartic acid. So GAU is going to code for aspartic acid, whereas if you shift that reading frame, CGA is going to, CGA is going to code for arginine. And that is going to change the resulting protein. So you have to be specific when we're looking at the 3-sequence codon. So your codon is found on the mRNA strand. An anticodon, important to write in your notes, is a set of 3 nucleotides that is complementary to the mRNA codon, and it's found on your tRNA, your transfer RNA, which we've learned about before. You can see it's kind of a triangle-shaped thing, and it carries the anticodon, and it also carries an amino acid. So if the anticodon is complementary to the codon, then it's going to bind and it's going to release that specific amino acid. So what you need to know here is the anticodon is found on tRNA along with the amino acid. Ribosomes consists of two subunits. You have the large subunit, which is the binding site, and then the small subunit, which is going to bind the mRNA. And this will make more sense when we talk about the process here in a little bit. Okay, so for translation to begin, tRNA is going to bind to a start codon and signal the ribosome to assemble. So once the mRNA is out in the cytoplasm, tRNA is going to bind to a start codon and it's going to allow a ribosome to assemble around that mRNA strand. And you can see that this is occurring at many spots. It's not just one ribosome. You can see many ribosomes binding around the start codon. So what happens is a complementary tRNA molecule binds to the exposed codon. So they kind of like glue together for a little bit, and it's bringing that amino acid close so that it can be released. So if we have AUG, we can see the start codon down in the bottom left corner of this picture. tRNA comes in with the complementary anticodon, which is UAC. And when it binds, it brings it close enough so that it can release that amino acid. As soon as that is happening, the next tRNA comes in, binds to the complementary codon, anticodon and the codon bind together. They're complementary to each other. They get close enough to each other, which then they would release the next amino acid. The ribosome is going to form a polypeptide bond between two amino acids. So you can see in picture number 2, between the two amino acids, there is that line, that bond. And you will continue to get bonds, peptide bonds, between those amino acids. So the ribosome will pull that mRNA strand just like a tape dispenser. When you're pulling tape, it's the same kind of concept. It's continually to pull out amino acids and binding them together with peptide bonds. Once that amino acid has been released and the peptide bond has secured the two amino acids, that tRNA, that transfer RNA with the anticodon, leaves the ribosome. The next tRNA comes in with a complementary anticodon. It's binding to the codon. It's going to then release the amino acid, and the peptide bond is going to form. This will continue until a stop codon is reached, and the ribosome will then release that protein and disassemble from the messenger RNA. Okay, so I'm going to go back here real quick and just one more time. This first picture is really good to remember because tRNA will continually come in. Anticodons and codons have to bind together. They have to be complementary to each other. A's and U's, C's and G's. When they bind together, the amino acid gets released. A peptide bond forms between those two amino acids. And then when that happens, the tRNA leaves the ribosome. The next tRNA comes in, binds, and like it shows in this picture, this right side picture, you can see 5 amino acids bonded together through peptide bonds. This continues until, again, the stop codon is reached. The ribosome then will release that protein. It will go and do whatever it has to do in its body, and the ribosome disassembles from the mRNA strand. Your teacher will take the time to also talk through this process. You'll do some practice with review questions. You'll take a walking quiz. You'll watch some more videos. So take the time to learn this process and ask questions if you have them.